Okay, I want to move on because time flies, and this is a really interesting thing which I'd like to describe and hopefully get some help with. Okay, if you had this knowledge, so here we go. We know where the people are living, right? We've got census data, maps, stuff like that. And we know geography. We know where the roads are, where the beaches are, where the ridges are, where the rivers are. And we know the rainfall, and we know the temperature, and things like that. If we also know where the mosquitoes are, right, then the scientists, particularly ones that are focused on mosquito-borne diseases, can run a big calculation. They call it a model. They model important parameters that help them estimate where future hotspots might be. So to give you an example, I'm sorry that the thing is so low, but as you can see, it's a map of Oahu. This work was done at the University of the University of Hawaii in Manoa by this group here, Becky, and they basically took that information and they used mosquito distribution data, this stuff, right? They had that data when they ran that analysis in 2011. They had it for Oahu. And they were able to run this calculation. Now, the number that they calculated is a little bit abstract, the so-called vector-to-host ratio. Basically, it tells you how many mosquitoes per person there were in a given area. And the idea is that if you're in an area with a high ratio, a lot of mosquitoes per host, then that host being human, then that host has a much higher risk of exposure to dengue or another virus than if it's a low ratio. Now, the thing about this is the kind of information that a vector control agency, a mosquito abatement district, whatever you want to call it, the people who are interested in fighting mosquitoes when an outbreak occurs, this is the kind of information that's very valuable to them. For instance, suppose last fall the Department of Health had a map like this for the Big Island, and it showed where the hot spots were. They could have gone in, done some really good abatement in those areas, and perhaps we never would have had an epidemic. So, the obvious question, what about the Big Island? Well, this is the problem. We don't have the data. You see, you can't take a calculation like that and do it unless you have good data going in. Remember the old computer adage, garbage in, garbage out? The problem is, we don't have that top left corner. We don't have the mosquito distribution data. All we have is data that is incomplete and at least 5 years old. So this professor who is part of that group, Durrell Kaplan, he's now in California at the California Academy of Science. He created a project, so he's still working on this issue. He created a project where citizens like us can take part. We can collect that data that's missing, because to collect the data requires a lot of time on the ground. A lot of, one entomologist in the back can't do it on his own. But it is something that we can do, and it's really not that hard. So let me show you a preliminary calculation that was done. Now this is a theoretical calculation of where they think the habitat is appropriate for Aedes aegypti. And the reason we're interested in Aegypti is because we know it's the more competent vector. So as you can see, there are some very familiar red areas, red being high probability of Aegypti being present. So the challenge now is to do this modeling and get meaningful answers out of it. We want to have meaningful data to go in it. And so the project, Mosquitoes in Hawaii, at inaturalist.org, is collecting data. We're up to about 90 observations now. We have lots of I think there's like 11 or 12 members. The problem that there is that the members at the moment, we have very few. Let me just go one more. Here's the data we have at the moment. The gray marks are historical data, and the red ones are recent ones. And you'll note that a few of them are out in the middle of the ocean. That's for privacy. The actual location has been obscured for the public, but the researcher can still know where it actually was. So you can see that over where all the red was, we don't really have any data yet because we don't have any volunteers. And what I would like to encourage people to do is to join the project and catch mosquitoes and observe them and try and identify them and submit data. So for instance, Don, here's your data. This is a picture, beautiful picture of, see the harp, the crescent lines, the arc lines on its back? That's an Egypti, very clear record. And he got it up there somewhere in that square box. Right? That's really an important data point. And we need many more of those. What we need is further down. This is one, you can't see it because of the room lighting. That's another Egypti. And you can see that's down in the Kalapana area. Right? You might not expect, the classic Egypti habitat is a very dry area. And we all know that Puna is pretty wet, although it's fairly dry down near the water. So that was someone who took a picture, and what you do is you use your phone. Literally, you can take those. The picture here was taken with a phone, and he caught it with a wine glass, right? And that's a wine glass you can see there. And he took the picture through the wine glass. So you get a clip-on lens for your phone, and the phone has the advantage that it tags your photo with your GPS location. So when you load it, it automatically knows where you took the picture. And it's relatively straightforward. It takes a little practice to be able to take pictures of mosquitoes. It helps to have tweezers, but it's something that anybody can do. And I really would like to encourage people to take part. Here's one I took, Albopictus. You can see the line very clearly. It's on the Red Road in Lower Puna. Catching mosquitoes is pretty straightforward. We have nets. Durrell sent a bunch of nets. Whoa. Great net. Nice, bright colored. I would love to give these to some volunteers. We have lenses like this for people who are interested in taking part. And has anyone got any questions?
Oh, but I have a couple of comments.
Sure.
If they're swarming around you and you really want to be killing them, if you wet your hands, your chance of hitting that mosquito will be about 99% more than if you have dry hands.
Wow, I want to repeat that.
It's so amazing.
Sorry, I don't know your name.
Barbara.
Barbara, okay, your comment that if you wet your hands, you're much more likely to be able to catch the mosquito.
Yes.
Than otherwise. I think that's a great idea.
And also, if one is biting you, if you can grab the opposite side, like on your arm, and pull it tight, he cannot escape because you have caught his stinger in your arm. You need a third person, a second person, like, okay, get him. I'm holding for you now. Okay. Get him.
You're hired.
Did you know that?
You're hired. So I have to warn you that these people who get really into this mosquito stuff, like John, so the picture of the calculation for the Big Island was done by a guy called John Winchester, who is a student of Durrell Kaplan in Manoa in 2011. And he came to visit a few months ago to help me put traps out in the Kalapana area. And, we were in this one area where there was a lot of mosquitoes around. He's just standing there. And I'm fighting him off, and he's just standing there. And then one lands on his arm, and he just watches it, and he waits, and he waits, and he just grabs it. It's like, I'm sorry, I'm not going to do that. No, but I think, there's lots of great ideas for how to catch them. Yes?
I'm curious, you showed the picture of the hot spots on the Big Island.
Yeah.
And I would have thought they would go where there's lots of rain. Don't they need water to breathe? And why do they pick dry spots?
Yeah, I live there as dry as...
So they need water to reproduce. That's absolutely correct. But the Egypti has evolved a very clever survival method So basically, the ability of its eggs to survive for long periods of time mean that it can live in a dry area that gets rainfall very infrequently. And then when it does get that rainfall, the egg gets immersed in the water, it hatches in 12 days, and it's out there biting people and laying eggs for 5 weeks. And that has turned into a very successful survival strategy for that mosquito. Albopictus is not quite as good because they don't survive as long in a desiccated
Why
state.
wouldn't they be able to survive in a wet place?
Why would they not be able to survive in a wet place? Good question. So mosquitoes don't exist in isolation. So when there's a container, many mosquitoes can lay eggs in it. And what you get is you get competition between the different larvae within a container. And it turns out that the Albopictus are a much more aggressive larvae. And they're able to outcompete the Aegypti larvae. And so therefore, and this has been observed basically all over the world, when an established population of Aegypti is invaded by a population of Albopictus, typically the Albopictus will push out the Aegypti. Now that does not happen where the Aegypti are specialized survivors, like for instance in an urban environment, where Albopictus doesn't find enough vegetation to do well. So it's competition that causes it.
Is it also true that they cannot survive in water over 4 foot deep, like for the catchments?
The question is, can they survive in water over 4 foot deep? I see no reason why they can't survive.
I've read that in numerous places that they can't because they can't float back up in suspension to breathe, and so therefore they die.
I'm pretty skeptical, but maybe the entomologist, Chris, do you know?
Have you not read that?
I've heard that as well, but I think, you can't really say that it could never happen, but generally deeper water doesn't favor mosquitoes. But water catchment tanks are a unique situation. Oftentimes there's a screen that floats in the water and things like that, but generally huge, deep reservoirs or something like that aren't as conducive.
Maybe that's because of laying eggs on the dry part. Do they need a dry part to lay the eggs or something?
Well, the ram is always dry.
Oh.
But there are people who have looked at diving depths and things like that. So,
Yes.
And if I swat one on my arm and then take a picture of it,
Yeah.
can you do an ID from a picture?
It's not hard to
I'm
swat.
so realistic here. I don't want to.
Okay, so the question really has to do with how badly mangled can it be, and you still be able to identify it. So the marking on the back is it's made up of very small scales, right? And they're very easily rubbed off. So I have caught what I think were Aegypti near my house, like that. And when I tried to ID them, I just couldn't convince myself that I was actually seeing what I hoped I would see. And that is very easy to do. That's one of the benefits of a net.
So the project seems like it's pinpointing locations, but
Yes.
how do you determine mosquito density from that? Is it just
So,
if you
good
have
question.
a density of volunteers, then
Good
you
question.
can
So the question is, the project is identifying locations. So that's known as presence records. Is the insect present there? And should we be getting information like the density or the numbers? And the answer is, we would love that data, but to get it is a major enterprise and beyond the scope of what we as citizen scientists can do, unless you really become a fanatic. But yeah, of course it would be better to have density, but the calculations, the modeling, can actually get quite a lot just with using the presence data. Okay, another question? Yes?
Given the lack of data, because of all the resource cuts over the last few years, do we actually know for sure that it's Aedes aegypti that has been responsible for the bulk of the dengue infections rather than Albopictus?
So the question has to do with which mosquito was responsible for the bulk of the dengue incidents. And I think I would have to defer to the Department of Health, but I'm pretty sure they'll tell you they don't know. Chris?
I'm just getting on board, but I know the CDC entomologists and entomologists from Honolulu are actively working on that. They collected a lot of samples. They're doing DNA testing. So they should have a better answer for that soon, forthcoming.
And Albopictus is across all the islands now, right? But Egypti is only here so far. Is that right?
Correct. It was eradicated. So Egypti was eradicated from the other islands 30 years, 40 years ago, a long time ago. And they didn't get it done on the Big Island, unfortunately. Yes, Tom?
More of an observation than a question. Karen put us in touch with your project a few weeks ago, and it sounded like it'd be fun. And the first time I tried it, I got some pictures that were unidentifiable. We passed them back and forth through emails, and we just couldn't determine what they were. And apparently, the samples turned dark fairly quickly. So, like, I put them in a jar and put them in the freezer, and then I got the camera set up a couple days later, and you just couldn't tell what they were.
Yeah.
And so, if you like immediately kill them and immediately photograph them, they're much easier to identify. So I just put them in a jar with some ethyl acetate, which is fingernail polish remover, and just straight away.
It'll kill anything.
Yeah, so Don's comment is about how when you catch some mosquito, a lot of times it will darken. And it's darkening because of the enzymes in the dead insect continue to work away, and they basically change the coloration. One trick I've used is to catch them and rapidly put them in very hot water, close to boiling but not boiling. Just dump them in that for a few seconds, and that, I read, slows down or breaks up the enzymes so they don't work, and actually I think that works. But it is definitely best if you can identify them pretty much right then. Okay, the question at the back first.
So like if one lands on your arm and you've got a good view of it, like if I have my glasses on, am I going to be able to see the little white crescents pretty
Before
clearly? I mean, or do you need a magnifying glass?
So do you need a magnifying lens to identify them? Probably. Yeah, you probably do need a magnifying lens, but for sure there are people who can look at them and see. John Winchester is holding that bug on his arm, and he says, "Oh, it's an albopictus." He could tell.
One that's with stripes, that's a different one?
They both have stripes on their legs, and they both have stripes on their lower body, their abdomen. But what we're looking at is the back of their back, behind their neck, the thorax it's called. That's where the really distinguishing feature is. Okay. Can I just go on one more slide? Because catching mosquitoes and making sure you don't crush them is kind of a pain. You can spend a lot of time and get one or two. So what I found was that it actually was really easy to go to places, cemeteries for instance, check the flower vases, find loads of larvae in them, collect the larvae, take them home, put them in peanut butter jars, and rear them out. So here's an example. These are, does anybody else find this picture creepy? They're staring back at me.
What's the magnification?
I don't know. This is about, I'd say, 3 or 4 millimeters. But yeah, it's relatively easy to rear them. You just simply put them in water and put a lid over it so that they can't escape when they hatch. And then you get to watch them. Sometimes you catch them and they're very small. You watch them get larger. These are quite mature ones, almost ready to turn into pupae. Pupae are, I kind of describe them to people as little shrimp. They're little round things and they zoom all over the place. They're very active. And the pupae are what hatch into adults. And they'll rise to the surface of the water, and then you'll often be able to see them half hatch. When they eventually do hatch, you can catch them in a net, or what I like to do is put sticky paper on top of the jar so they have nowhere to go, and then they end up landing on it, and then you can just pick the paper up, take a picture of them, and then get rid of them. That works really well.
Wait, I don't understand. You
Okay.
put the sticky paper underneath the jar?
Oh no, I'm sorry.
Lid?
Under the
You
lid.
take the lid off and you put the sticky paper on top where the lid was.
So they're not going to fly away?
So they can't fly away. And I usually poke a hole or two in the paper that they can't get out of. And they don't like it. It's very clear. They'll go up to it and they'll fly away and go up to it and fly away. But eventually they'll land on it and they'll stick. Yes.
When they hatch, it's going to be a mixture of males and females. Do the
Correct.
males look different than the
Yes, they
females?
do. I wonder if any of these pictures back here, they're not quite the resolution. The key character, maybe this one, I can't tell. This might be its antenna. Basically, you tell the difference because the males have very fluffy antennae, and the females don't. That's how you tell the
But
difference.
they'll have the same markings, so you can tell the
Yep,
species.
that's right. Yes.
You said that they eliminated the Egypti on all the other islands.
Right.
How'd they do that?
Oh, DDT.
I used to live on, in the 50s, they'd drive trucks around and spraying in the air, like through our neighborhood.
Yeah, I think it was DDT. They basically, yeah. But remember this, Gypti are uniquely adapted to humans and living with humans. So that means that they're not in the jungle. You're not going to get bit by an Egypti if you're out in the jungle in the middle of nowhere. You're going to get bit because they like living in your house, they like living around your house, and they like breeding in the containers that we leave. So Egypti are actually probably the easiest mosquito to get rid of. It will be harder to get rid of albopictus.
It's interesting that the outbreaks on this island, the big places on this side of the island and on the other side, were basically uninhabited areas at beach parks.
So people live there, but people also visit there.
Yeah.
So, for instance, back in
That's
that
right.
modeling, the observation was, if I could just repeat it, was that the outbreaks here happened in areas that are not heavily populated. So that's a very accurate observation. And if you look at, so the researchers doing that modeling, they take that into account. They have a data set that shows where people live, and then they have a data set that shows where people recreate, where they go out and hang out on the beach. And they survey people to find out what percentage of people hang out on the beach and so on. So it can all be accommodated, yes. Yeah, it's a difficult question. I don't know why the outbreaks were there, except the large transient population.
What about Zika? Is that carried by any of these?
Both of these carry Zika, yeah, unfortunately.
Others also?
Say again?
Other mosquitoes also?
As far as I know, those are the only two on this island that carry it. Yeah.
The fly sticky traps that you pull out, would they work as far as just catching them with, is there an attraction of smell? Have you ever tried that one? Or?
Question about fly paper, I don't know the answer. I haven't tried.
Of putting one of those down in your bait thing just to...
Using that? Oh, well, so the question, this is an interesting question related to traps. I am proposing using poisons. It absolutely is not the only way of doing it. In fact, the CDC has a trap that uses a sticky surface that they catch them on. No chemicals. That's great. You know, you can put that on an organic farm. And I know that the company that makes the trap and kill, SpringStar, that make the commercial trap, they are close to releasing a version that will also be a sticky trap. And that will be, again, real nice for people interested in organic farms and so on.
Are the SpringStar traps similar to what you showed us? Or?
Well, so the question about SpringStar traps. There, let's look at the trap. Oh, it's empty. Thank you. Okay, so here we have a black container, and it has smooth walls, right? Really, the smooth walls in this case are not so important. The reason being, they have a strip here, which doesn't have any poison on it. And the strip is going to just be stapled to the side, and it provides a place for the mosquitoes to hang out and maybe lay eggs as well. It's very common to find eggs on the red. But instead of a contact poison like we use because it's easy to buy at the hardware store, they use, there's a little red thing in here, I won't open it, It's a little red pad that you staple to the wall, and that pad emits a vapor. So it's a vapor phase poison. So it works quite differently. The attractant is still water. You fill this up with water. The water will have algae growing in it after a while, so it's attractive to the females. They'll fly in. It's a confined volume, so the vapor doesn't escape very quickly, which means it builds up in there, and they get a lethal dose and die from that. They work very well, and yeah, they've been deployed in many places successfully. Yeah. Did that answer your question? I forget what your question exactly was.
About the sticky trap.
Oh,
You
how
can
they
buy
get
if you put, along with your stick in there, if you put one of those, the possibility of them flying and getting stuck on that is.
Right.
The water that you use in the trap can't have any chlorine whatsoever? I mean, because
Well-
water here is chlorinated.
You sit it for a
So
few days and it evaporates.
the researchers found that chlorinated water acted as a repellent. That's I don't have any experience of that, but that's what I read.
Yeah, pond water.
Yeah, the chlorine will evaporate eventually, but it will take time. And you might as well just start with pond water, or water from a mud puddle, or from your bromeliads.
Or you put it in the sunlight, that degrades the chlorine.
So,
Sure.
do mosquito fish or the little guppies, do they really work for
So the mosquito fish,
water
question is about
garden?
mosquito fish for large containers.
A water garden.
I looked into the issue of catchment tanks and because it's an important issue for those of us who are not on county water. What I found was that the EPA doesn't really have any good recommendations. So I looked to the World Health Organization. Their number one recommendation was guppies. It was kind of charming. They said that these are very successful in villages because the children love them.
Can you volunteer to have, to just have monitoring traps on your property and have a point person come and into an area and collect and identify?
So, yeah, you can do that. Yeah. So using traps to monitor for presence is quite a bit of work. Somewhere I have one. This is what they look like. It's a design out of Brazil. It was given to me by Capon and Winchester. John brought them over. Put water in the bottom here. Inside the tube is sticky surface. The mosquitoes fly in through the cone, and they get stuck. The problem is that you need to check it about every 4 or 5 days, because any longer than that, and the mosquito kind of turns into mush, and you can't identify it. It's quite labor intensive to use traps like this. If you want to, I'm absolutely happy to provide you with some. Yeah, it's possible to use traps, for me personally, larvae is the simplest way to go, and then catching them with a net or by hand. Yes.
Is this presentation available on your website?
Will be. Yeah,
Okay.
it will be.
I mean, Rod's going to have it on the website for
Yeah, sure,
Science Cafe.
sure. Yes.
What are your thoughts about establishing a mosquito abatement district for the county of Hawaii?
So the question of establishing a mosquito abatement district, I think I'll probably punt that question.
Okay.
It's a political question.
There's a lot of politics there.
Question at the back.
The genetically engineered mosquito that they released recently in Florida, is it Aedes aegypti?
I am not sure. I believe it is,
Yes,
but
it
I'm not
is.
sure. Thank you. So the answer is yes, it is. The question was the genetically modified mosquito they released in Florida was aegypti, yeah. I believe they're working on albopictus ones as well, but I'm thinking China.
These are the sterile males.
Yeah, there's a lot of cutting edge biology research going on in this domain, and it's hard to keep up with it. Yeah.
So Dr. Hemi, in February when he was here for the community meeting in Pu'uhonua, Ohana now, told us that the outbreak, he thought that the outbreak was driven by albopictus primarily. But maybe they didn't have enough data on the other clusters of the Egypti. But didn't Durrell Capon, did he do a survey in Ocean View? Or somebody did a survey in Ocean View in 2011 and they really found Egypti?
Let's see if the data happens to be in the, oops, wrong way. Oh, by the way, we talked about the adulticides and larvicides. I put a table with a whole bunch of information in it for you to refer to later, including the research papers that they come from.
That's on the website?
Yep. Oh, come on, come on. Here we go. So there's some records down there. But yeah, I'm afraid I don't know. Sorry.
We'll wrap it up.
Sounds good. Thank you.